Hello everyone, in this video I'd like to present you my PhD thesis. In my thesis I focused on the methodology of eye tracking. Eye tracking is a useful tool which could be used in many research fields to obtain quantifiable data about where people look on the screen. One type of the eye tracker is visualized on the right, while the subject is viewing the screen. The blue dot in the video shows a position where the subject looked during the trial. Here's an example of typical workflow of an experiment that uses eye tracking. First, we present an image. Then we measure eye movements during the task. The blue dots show several positions where the participant looked. Eye trackers usually sample at frequencies between 250 and 1000 Hz. Therefore, we have several thousands of data samples, even for small presentations. Finally, there are two main types of analysis. Either we define areas of interest and compute for how long did the participant look at a particular location, or we compare the whole distributions of eye movements. The workflow that I just described dealt with static stimuli. The situation is more complex in the case of dynamic stimuli. Movie is one particular example of dynamic stimuli. In dynamic tasks, the eye movements are usually represented as scan patterns. Scan patterns are spatial temporal representation of eye movements, and here you can see an example of such scan pattern. So far described approach worked with static stimuli. However, for dynamic stimuli, such as movie clips, the situation is more complicated. Although we could use areas of interest, we have to specify them frame by frame. As the content of the movie changes every frame, we need to work with timescale. The temporal dimension is often omitted in static tasks. There are still some unresolved questions regarding analysis of scan patterns, on which I focused in this thesis. As the movie clips contains a lot of content, it is hard to formalize it using a model. Therefore, I decided to use a different dynamic task called multiple object tracking. Here you see an example of such task. Your goal is to track several moving objects at once. The green objects are targets, while the gray objects are distractors. After a while, all objects become indistinguishable and start to move. After a while, they stop, and subject task is to select the tracked targets. In this thesis, I focused on the three main goals which are related to the analysis of the scan patterns in dynamic tasks. First, how robust are the metrics that are used for comparing scan patterns? How do those metrics relate to each other? How sensitive are the metrics to the systematic noise in the data? Second, if we compute similarity between scan patterns, how could we test that the differences between groups of scan patterns are significant? Finally, can we predict scan patterns in MOT task? If we could formalize eye movements in this task, we could get a baseline for further studies regarding scan patterns. Let's look on the first question: how robust are the metrics for comparing similarity of scan patterns? There are many metrics that could be used for scan pattern comparison. There were several published studies comparing the robustness of each metric for static tasks, but none of the studies for dynamic tasks. I explored how are these metrics sensitive to the noise. I created 4 simulations. First 3 were preparatory for simulation 4. In this simulation, I selected 5 metrics that can be used for scan patterns in dynamic tasks and measured their scalability with added noise in the scan patterns. I selected 3 transformations: translation, rotation and scaling, and applied 1 or 2 transformations at the same time at each scan pattern. Then I computed the distances between the original scan pattern and modified one for each of the metrics. Here are the examples for each of the transformations. Distances between scan pattern evaluated by each of the metrics were highly correlated with each other. Here are examples of contour plots which show how the metrics scale when we apply two transformations at the same time. Here you can see scaling and rotation. Contours denote same level of distances. I also developed an R package that is publicly available for other researchers, so they can get similar benchmark comparison for different metrics and different transformations. Regarding the second goal, so far I talked about the methods for measuring distance between scan patterns. But how should we proceed when we want to test whether this distance is significant? If we have two groups of scan patterns, here denoted as G1 and G2, the question is whether the scan patterns in one group are more similar to each other than to the scan patterns in the other group. In other words, is the within group variability statistically different from between group variability? So far, there was only one solution to this problem by Feusner and Lukov. Using their method, I computed distances between all pairs of scan patterns in each group and also between all pairs of scan patterns between group. By subtracting the average of both between group and within group distances, I obtained the d-star value which represent the current grouping. Finally, I replicated the computation for additional grouping and compared the original d-star value with the distribution of other average distances using permutation test. I extended the original method for metrics that compute the distance between whole groups of scan patterns, and therefore it removes the necessity to perform all pairwise comparisons, and also created another method that tests the differences from another perspective. I evaluated the performance of the three metrics using artificial scan patterns that were created as a random walk process. Both of the novel methods outperformed the original strategy. I showed the practical application of the use of the best strategy in two experiments regarding scan pattern symmetry. We are getting to my third goal, how we could predict eye movements in MOT tasks. I assembled data from three behavioral multiple object tracking tasks and trained several neural networks to predict the eye gaze position during the trial. The performance of the trained networks was evaluated using median of Euclidean distances between eye gaze and predicted values for each frame. I also computed several analytical models from previous studies as current state-of-the-art models. The neural network models outperform the current analytical models by 10%. Below, you can see predictions for one selected frame. The red diamond is real eye gaze, the diamonds of other colors are predictions from models, the blue diamond shows the prediction from the best neural network model. That is all. Thank you for your attention. All the materials and the scripts are publicly available on Open Science Framework, and our package for creating additional simulations is on the GitHub. Read the full thesis and don't hesitate to contact me if you have any questions.